Imagine a cure for cancer, not a treatment, not a way to manage the disease, a cure. Now, imagine this cure comes not from a billion-dollar laboratory in Switzerland or a sprawling corporate campus in New Jersey. Imagine it comes from a dusty, sun-baked corner of West Texas, and it is derived from a simple, humble, and profoundly misunderstood cactus. You are picturing the life's work of Dr. Alistair Finch. Now, imagine the door to his small, independent laboratory splintering open. Imagine the flash of black uniforms, the glint of firearms, the cacophony of shouted commands. Federal agents, DEA. They are not there to celebrate a breakthrough. They are not there to secure a world-changing discovery for the good of humanity. They are there to shut it down. They are there to seize his research, confiscate his samples, and brand him a criminal. This happened in 1989 in the United States of America. The official story is one of regulations and controlled substances, a simple, unfortunate bureaucratic mistake. The real story is far more complex. This is the story of a man who saw what no one else could, the story of a system that crushed him for it, and the story of a truth so powerful it could not be buried forever. To understand the tragedy of Alistair Finch, you must first understand the world of 1989. The Cold War was ending, but a new war was at its zenith, the war on drugs. It was a time of moral panic, of zero-tolerance policies, and of immense federal power being brought to bear on anything even tangentially related to illicit substances. The evening news was a nightly parade of drug busts, seized assets, and stern warnings. It was a climate of fear. And in a climate of fear, nuance dies first. Simultaneously, the war on cancer was being waged on a very different front. It was a war of attrition. The primary weapons were surgery, radiation, and chemotherapy. Think about that last one, chemotherapy. It was, and often still is, a brutal, scorched earth campaign inside the human body. A chemical poison designed to be slightly more toxic to cancer cells than to healthy cells. It was a numbers game, a gamble. Patients were ravaged. Their hair fell out. They were racked with nausea. Their immune systems collapsed. They suffered, profoundly, in the hope of buying more time. It was the best they had, and it was a multi-billion-dollar industry. The research, the manufacturing, the administration, entire corporate empires were built on these cytotoxic compounds. Companies like Pfizer, Merck, Bristol-Myers, giants of industry, with legions of researchers, armies of lobbyists, and stock prices that hinged on their next patented blockbuster drug. Their approach was monolithic. Find a new synthetic molecule, patent it for billions, and sell it for hundreds of thousands of dollars per patient. The system was entrenched, powerful, profitable, and into this world walked Alistair Finch. He was not a product of this system. Born in 1942 in Terlingua, Texas, a ghost town clinging to the edge of Big Bend National Park, Finch was a child of the desert. He was not raised in a sterile suburb or a bustling metropolis. He was raised in a world of harsh realities and breathtaking resilience. His playground was the Chihuahuan Desert. While other boys his age were collecting baseball cards, Alistair was collecting specimens. He was fascinated by the things that survived where nothing should. The ocotillo, which could look dead for years before bursting into flame-red blossoms after a single rain. The creosote bush, which poisons the ground around it to eliminate competition. And above all, the cacti. He saw them endure unimaginable heat, months of drought, and freezing winter nights. They were nature's ultimate survivors. His father, a retired mining engineer, saw the boy's obsessive curiosity. He didn't try to stamp it out. He encouraged it. He taught him the fundamentals of chemistry and biology, using a battered college textbook. In the dusty garage of their small adobe home, they built a makeshift laboratory. At 12, Alistair successfully extracted and identified the specific alkaloids in a dozen local plant species. At 15, he built his own gas chromatograph from salvaged parts. He was not a normal child. He was a prodigy, forged in solitude and sunlight. He earned a degree in botany and chemistry from Sul Ross State University in Alpine, a small college known more for its rodeo team than its science program. He didn't go to the Ivy League. He didn't want to. He felt the answers weren't in their libraries. They were out in the desert, waiting. His obsession found its focus in 1975. His mother was diagnosed with pancreatic cancer. He watched as the disease and the subsequent chemotherapy ravaged the strong, vibrant woman he adored. He saw the suffering, the loss of dignity, the slow, agonizing fade. She passed away in the spring of 1976. Alistair Finch was 34 years old, and he had just found his war. He looked at the towering, complex world of oncology and saw a fundamental flaw. Everyone was searching for a bigger hammer to smash the cancer cell. More potent poisons, more focused radiation. They were asking the wrong question. The question wasn't just, how do we kill cancer? The question was, why does cancer live when it should die? Let me explain. Every day, in your body, cells make mistakes when they replicate. Most of the time, the cell's internal quality control detects the error and triggers a process called apoptosis. Programmed cell death. It's the cell's beautiful, elegant, and essential act of suicide for the greater good of the organism. It's not a violent death, it's a quiet, orderly disassembly. Cancer, in its simplest form, is a cell that has forgotten how to do this. Its apoptosis trigger is broken. It achieves a horrifying form of immortality, replicating endlessly, ignoring all the signals to stop. Finch's idea was radical. What if, instead of carpet bombing the body with poison, you could simply remind the cancer cell how to die? What if you could find a key to fit the rusty lock of its apoptosis pathway? It was a breathtakingly elegant thought, and it was completely outside the mainstream. The big pharmaceutical companies had no interest. You can't patent a biological pathway. You can't make billions from a reminder. They needed a new patentable molecule, a hammer. Finch knew the answer wouldn't be found in a corporate lab. He returned to his first love, the desert. He reasoned that the same chemical defenses plants use to survive extreme stress, compounds that regulate their own growth, fight off invaders, and manage cellular repair, might hold a clue. He began a systematic study. For years he worked in near-total obscurity, funded by a small inheritance and the occasional grant from local Texas foundations, who were more interested in supporting a fellow Texan than in understanding his esoteric research. He tested hundreds of compounds from dozens of species. Yucca, agave, mesquite. Nothing. Then he turned his attention to a particularly controversial plant, Lophophora williamsii, peyote. He wasn't interested in its hallucinogenic properties. That was a distraction, a side effect. He was interested in its chemistry. This was a plant that produced an incredibly complex array of phenethylamines and isoquinoline alkaloids. It was a chemical factory, honed by millennia of evolution. And it was a Schedule I controlled substance, the most restricted category of drugs in the United States. To the DEA, it was in the same class as heroin. Finch, through his academic connections and sheer persistence, managed to secure a rare federal license to study the cactus for legitimate research purposes. It was a mountain of paperwork, a bureaucratic nightmare. But he did it. Inside a small, temperature-controlled greenhouse behind his lab in Alpine, he grew his own specimens. He began meticulously isolating compounds, one by one. The primary psychoactive component, mescaline, was discarded immediately. He was looking for something else, something hidden. In the autumn of 1987, he found it. It was a complex isoquinoline alkaloid he provisionally named apoquinine for its apparent effect on apoptosis. It was present in trace amounts. In its natural state, its effect was negligible. But Finch was a master chemist. He discovered a simple enzymatic process to modify the molecule slightly. He cleaved off a specific methyl group, a tiny, almost insignificant alteration. The result was a new, stable, and non-psychoactive compound. He called it Finch 17. It was the 17th major variant he had synthesized. The eureka moment came on a Tuesday morning in October. He introduced a solution of Finch 17 into a petri dish containing a virulent strain of human glioblastoma cells, a notoriously aggressive brain cancer. He watched through the microscope. For the first hour, nothing happened. The cancer cells continued to divide, a chaotic, uncontrolled bloom. Then he saw it. A single cell, on the edge of the colony, simply stopped. It ceased its frantic division. It began to shrink. Its membrane blebbed, forming small, bubble-like protrusions. The nucleus condensed. It was not exploding. It was not dissolving in a wash of poison. It was neatly, cleanly, and quietly disassembling itself. It was undergoing apoptosis. Within hours, the entire Petri dish was clear. The cancer was gone. He repeated the experiment with healthy brain cells. Nothing happened. Finch 17 was completely inert in healthy cells. It only affected the cancerous ones. It was the key. The key he had been searching for his entire life. A key that only fit the lock of a broken cancerous cell. This was not a bigger hammer. This was a ghost key. He moved to animal trials. Mice, implanted with human tumors. The results were miraculous. A single, low-dose injection. The tumors didn't just stop growing. They vanished. They melted away in days, with no discernible side effects. No hair loss, no weight loss. The mice were healthy, active, cured. An absolute triumph. Finch meticulously documented his findings. The chemical structure, the synthesis process, the results of the in vitro and in vivo tests. On January 9, 1989, he filed for a United States patent. Patent application number 7334918. He also submitted his research to the New England Journal of Medicine and the Journal of the American Medical Association. He had done everything by the book. He knew he was on the verge of changing the world. But Alistair Finch didn't know that his real battle was just beginning. The first sign of trouble was the silence. His submissions to the medical journals were rejected. Not after peer review. Just rejected. The form letters were curt, dismissive. Topic not of sufficient interest to our readership. Not of sufficient interest? A non-toxic cure for glioblastoma? It made no sense. Then, in March, he got a visit. Two men in dark suits. They weren't from a university or a research foundation. They were from the legal department of a massive pharmaceutical corporation. He never named them publicly, but his private notes suggest it was a company whose entire oncology portfolio would be rendered obsolete by Finch 17. They didn't threaten him. It was worse. It was condescending. They reviewed his data with polite skepticism. They called his results anomalous and likely unrepeatable. They spoke of the immense cost of clinical trials, the regulatory hurdles, the complexity of the market. Then came the offer. They offered to buy his patent application, not for a partnership, not to develop the drug, to buy it. The price was insultingly low, a few $100,000, less than the cost of a single television ad campaign for their current leading chemotherapy drug. Finch understood immediately. They didn't want to develop it. They wanted to bury it. To put his ghost key in a lead-lined box and drop it to the bottom of the ocean. He showed them the door. That was his mistake. He thought he was in a scientific debate. He was actually in a knife fight. 2 months later, the DEA came. The raid was on May 17, 1989. It was a full-scale operation. A dozen agents in tactical gear. Local sheriffs providing backup. They treated his small lab like a cartel compound. The warrant was based on an anonymous tip. A tip that Dr. Alistair Finch was not using his Schedule I license for research, but was instead manufacturing and distributing mescaline. It was a lie. A complete fabrication. But in the zero-tolerance climate of 1989, a tip was all they needed. They smashed his door. They ransacked his lab. They were not careful. They were destructive. Delicate glassware was shattered. Years of notebooks, filled with meticulous observations, were thrown into evidence bags, their pages jumbled. His computer, containing all the digital data from his experiments, was seized, and his samples, all of them, the petri dishes, the cryovials containing his precious purified Finch 17, the mice that were living proof of his cure, all were confiscated as evidence. They even ripped his licensed peyote plants out of the greenhouse. He was arrested. photographed, fingerprinted. He spent a night in the Brewster County Jail, a 67-year-old botanist and chemist, locked in a cell with men half his age, accused of being a drug kingpin. The charges were, of course, baseless. There was no evidence of mescaline production. He had followed the letter of the law on his research license. After 6 months of legal wrangling, all charges were dropped. But the damage was done. His reputation was in ruins. The local papers had run the story of the raid on the front page. The story of his exoneration was buried on page 12. His lab was a wreck. The seized equipment was eventually returned, but much of it was damaged. His research was gone. His notebooks were returned, but they were out of order, contaminated. The seized samples, the vials of Finch 17, the living, cured mice, they were never returned. They were destroyed as hazardous biological and chemical materials, according to a boilerplate government form. They had not just shut him down. They had erased him. He had lost everything. His life's work. His savings spent on lawyers. His good name. He was a broken man. Before continuing, what do you think? Do you believe the raid was a tragic bureaucratic overreach fueled by the hysteria of the era? Or was it something more sinister? A targeted strike, orchestrated by powerful interests who saw a multi-billion-dollar industry threatened by a simple cactus and an old man in a Texas lab. Let me know your thoughts in the comments below. I read them very carefully. And if you believe stories like this need to be told, please take a moment to press the like button. It tells the algorithm that this history matters, and it helps ensure more people see it. Now let's continue. The years after the raid were dark for Alistair Finch. He tried to rebuild. He tried to recreate his work, but the spark was gone. He was in his 70s. His funds were exhausted. The scientific community, which had once been merely indifferent, now saw him as a pariah. A crank. The peyote cancer cure guy. He became a recluse. He sold his property in Alpine and moved back to the tiny, dusty town of Terlingua, to the house he grew up in. He lived quietly, surrounded by the desert that had once been his inspiration, but now seemed to mock him with its silent indifference. The world moved on. The war on drugs evolved. The war on cancer continued its slow, brutal, and immensely profitable march. New chemotherapy drugs were developed, each one hailed as a breakthrough, each one costing more than the last, each one carrying a devastating payload of side effects. Alistair Finch was just a footnote, a forgotten casualty, a cautionary tale whispered among independent researchers about the dangers of challenging the system. But a truth, a real fundamental truth, is a resilient thing. Like a desert seed, it can lie dormant for decades, waiting for the right conditions to germinate. The rain came in 2018. It came in the form of a young, ambitious postdoctoral researcher at Stanford University named Dr. Lena Petrova. Petrova was working on computational drug discovery. She was using artificial intelligence to sift through vast databases of abandoned patents and forgotten scientific papers, looking for promising molecular structures that had been overlooked. Her algorithm flagged a curiosity, a patent application from 1989, number 7-334-918. It had been abandoned after the initial filing fee was not paid. The molecule, a modified isoquinoline alkaloid, had a unique structure. The AI predicted, with 98.7% confidence, that it would have a high binding affinity for the apoptosis pathways in specific cancerous cells. Petrova was intrigued. She dug deeper. She found the name Alistair Finch. She found the newspaper clippings about the DEA raid. She found the dismissive rejections from the medical journals. She saw the pattern. And she didn't believe in coincidences. Working in her own time, on weekends and late nights, she synthesized the compound from the information in the patent application. Finch 17 was reborn. She tested it on glioblastoma cells, on pancreatic cancer cells, on lung cancer cells. The results were exactly as Finch had described 3 decades earlier. It was a miracle in a petri dish, a clean, targeted, non-toxic kill. Petrova didn't work for a small Texas foundation. She worked for Stanford, and her boss was David Chen, a Nobel laureate. When she showed him her data, he didn't dismiss it. He replicated it, and then he picked up the phone. What followed was a firestorm. Stanford, in partnership with a rival pharmaceutical giant, one that had not been involved in the 1989 incident, fast-tracked the development. Because Finch's patent was abandoned, the compound was in the public domain. They tweaked it slightly, gave it a new name, proapoptin, and filed their own patents on the new variant and its delivery method. The story broke in early 2019, the 30th anniversary of the raid, the Wall Street Journal, the New York Times, 60 Minutes. The narrative was explosive. A forgotten Texas genius had cured cancer in 1989, only to be crushed by the government and ignored by the establishment. Now his work was poised to upend modern oncology. The corporation whose lawyers had visited Finch all those years ago found itself in the crosshairs of a massive public relations nightmare. Their stock plummeted. There were calls for congressional investigations. The anonymous tip to the DEA was traced back to a shell company with links to their former legal department. The evidence was circumstantial, but the conclusion was damning. In a desperate act of damage control, the company's new CEO did something unprecedented. He flew to Texas, he held a press conference, and he apologized. He apologized to the memory of Alistair Finch. He announced the creation of a multi-billion-dollar foundation, the Finch Foundation for Independent Scientific Research, dedicated to funding unconventional, high-risk, high-reward ideas that exist outside the corporate mainstream. It was a stunning admission, a public act of contrition from the heart of the impersonal system. But Alistair Finch was not there to see it. He had passed away peacefully in his sleep in 2015, at the age of 73. He died a broken and forgotten man. He never knew his vindication was coming. Today, when you hear about new targeted therapies for cancer, drugs that work with the body's own systems, you are hearing the echo of Alistair Finch's ghost key. Proapoptan and drugs like it are now saving lives. His story is more than a tragedy, it is a lesson. It demonstrates that genius is not confined to prestigious institutions. It can blossom in the most unexpected places. It proves that the most elegant solutions are often the simplest, and that nature remains our greatest teacher. But it also serves as a chilling reminder. A reminder that powerful systems, whether governmental or corporate, are not inherently moral. They are driven by their own inertia, their own priorities, their own survival, and they can be blind or willfully ignorant to the truths that threaten them. How many other Alistair Finches are out there? How many other cures for how many other diseases have been lost in a file, dismissed by a committee, or crushed by a raid? That is the question that should haunt us. Every time a disruptive idea is mocked, every time a lone voice is silenced, every time a patent is buried, we risk losing another ghost key. Alistair Finch's legacy is not just a molecule in a vial. It is a testament to the ferocious power of a single obsessive mind. It is a symbol of defiance against a system that values profit over progress. He fought the war, and he lost the battle. But in the end, his truth won. Alistair Finch, 1942 to 2015. The man who listened to the desert and found a cure for the darkness.